All right, everybody, welcome. If you're here for the asymptomatic COVID-19 testing webinar, you are in the right place. You're here with John Harrell from Lighthouse Lab Services and Kimberly Zunker from Acumen today. We're going to be talking about asymptomatic testing. We're going to be talking about clinical validation requirements. We're going to touch on some changes that are happening. News stories are flying. FDA regulatory oversight of LDTs is kind of up in the air, and there's a lot of chatter. So we'll try to start to address that here today as well. We'll eventually do a second session on that, and that will be your chance to hear a little bit more. We're going to probably do that on the 2nd of September, and so we'll push that out a little bit later so you guys can register, because we're still digesting all the information out there about that. But today we wanted to tackle asymptomatic testing. There's a lot of questions about this. You know, is it needed? How do you go about it? What's required? How do you treat it differently? So we do have a poll on the right-hand side of your screen that you'll see. I'll do a little bit of housekeeping here. You'll see a few different sections. We have a chat box area. That's more for if you have a question, technical issue or something, feel free to chat there. There's a Q&A section where you can post or respond to a question, and we'll try to keep those in a thread together so it makes sense as those different replies come in. And then we have a poll that's posted. Go ahead and take a look at that and respond if you're interested. We're asking if this is your first time hearing about the HHS announcement that rolls back the FDA's regulatory oversight of LDTs and what you think about it. We'd just love to hear from you guys some feedback on what the industry thinks. And there's a handout section. I think we will be dropping a handout in there on asymptomatic validations that you can check out and take with you. But let me introduce myself and tell you a little bit about us, and then we'll move right into the material. So Lighthouse Lab Services, we're a laboratory consulting firm. We specialize in building labs, recruiting and staffing for laboratories, and then doing consulting work that ranges from validating LDTs is something that we do a lot of, to getting labs licensed to receive samples from other states, et cetera. We did merge recently with Elite Diagnostics, so now we're one company with two brands. We're eventually going to be all under one brand and working towards that. But we continue to grow, and if we can help you with any of your needs, whether they're recruiting, staffing, consulting, laboratory directors, we supply the lab directors to 165 plus labs out there now. Let us know if we can help with that, and we would love to chat with you and just figure out what's going on at your group and see if there's something we can do to assist you in scaling. So I put something on the screen you can respond to if that's of interest. Otherwise, I did want to just touch on the elephant in the room. Was we developed this webinar to talk about the FDA's regulatory template and kind of what is required when doing asymptomatic testing, and maybe get into some of the validation requirements that were unique to that. And then there was a little bit of a curveball thrown to us at the end of last week. Trump administration barred the FDA from regulating LDTs. What does that mean for us? It does not mean that EUAs go away completely, and we're going to let Kimberly get into that a little bit more, but it does change the game a little bit. And so we're going to touch on it. Like I said, we will have a follow-up webinar on the 2nd of September to go into that deeper, and we're going to try to get some experts onto that webinar that will be able to answer questions. We've been watching the news and trying to hear response from some of the agencies, and that's been in short supply. So a lot of questions, not a lot of answers at this point. But let's get into introducing you guys to Kimberly Zunker. Kimberly Zunker's the consulting manager for Acumen. She's got 10 years of lab experience, both working on the bench and in leadership, and has worked her way up and sees the lab industry from all the lenses and perspectives. Has a really nice comprehensive view of that. And she's going to talk to us a little bit today about asymptomatic testing and what that means. So I'm going to go ahead and I'm going to hand things over to Kimberly.
Thank you, John. So good afternoon, everyone. Good morning if you're on the West Coast. My name is Kimberly Zunker. As John mentioned, I'm the consulting manager for Acumen. Acumen itself is a laboratory performance partner. Our consulting department works with labs all over the nation to answer their strategic business questions and meet their laboratory needs. Since March, my role at Acumen has really evolved to include a significant amount of COVID research. So today I'll be applying that research to learning and sharing more about asymptomatic testing. Here are our learning objectives for today. So we're going to start by discussing the recent HHS announcement. As John mentioned, we had to do kind of a quick pivot for this webinar in light of this announcement. Quite frankly, that's not exactly unusual in this COVID situation that we've all been living through for the past several months. It feels like it's been just one pivot after another, from supply chain constraints to shifting regulations, as we saw last week. So we're going to dive into that. We did add a slide specifically to go over what that announcement may or may not mean and any possible implications of that as we know them right now. We're then going to look to understand the difference between testing asymptomatic and symptomatic populations. Finally, we're going to look at the validation requirements under both the FDA as well as CLIA. So let's go ahead and get started. So late last week, HHS removed the LDT, or Laboratory Developed Test, Emergency Use Authorization requirement. They determined that the FDA could not require a pre-market review of laboratory developed tests absent notice and comment rulemaking. Emergency use authorizations for any laboratory developed test can still be submitted on an optional basis, but HHS has determined that the FDA can no longer require it. It is important to note that with this announcement, labs without an EUA or other pre-market approval would not be covered under the PrEP Act. The PrEP Act is what provides liability protections for labs performing testing in emergency situations like the COVID pandemic. At this time, we have not yet seen any FDA response to this announcement. In fact, I was on the FDA town hall just prior to this meeting, and they stated that they would not answer any questions about the HHS announcement. Therefore, we're simply not sure what kind of implications this may have on the EUA process. Additionally, under the current FDA guidance, labs can perform testing as soon as they're validated while waiting on their EUA approval. So it's not truly clear if this announcement is actually going to increase the availability of laboratory developed tests in the market or not. Finally, nearly every week on the FDA town hall, we have manufacturers that talk about the need for an EUA in order to sell their tests to laboratories. They relay that when they approach a lab without an EUA, the test is seen as subpar or low quality, and they're unable to sell it. This has resulted in many questions surrounding timeliness of EUAs because there is a need for the EUA as a market differentiator. So even though they're allowed to sell in the market without an EUA, often labs are simply not buying. At this point, the EUAs have become a marker for quality, especially after we've seen highly publicized issues with false positives as well as generally poor quality serology tests. So all of that has combined to create this market that does show a preference towards tests with an EUA. This market preference is likely to continue for laboratories developing their own tests. So there will most likely be a continued role for emergency use authorizations in asymptomatic testing. Currently, asymptomatic testing claims have only been approved in laboratory developed tests. So it is possible that manufacturers will begin validating their kits for use in asymptomatic populations, but for now, most asymptomatic testing claims are occurring within the laboratory developed test realm. So to be clear, labs have been able to technically test asymptomatic patients for quite some time. The FDA has effectively built in a loophole surrounding asymptomatic testing by stating that all COVID tests require a doctor's prescription so long as you have a doctor's order and your doctor has a reason to suspect that you may be positive for COVID, even though you're not symptomatic. Asymptomatic testing can still be performed. The biggest difference is that the test itself cannot claim that it can be used or make any claims surrounding performance of testing in the asymptomatic patient population. So why is that important? Why do we need those claims for the asymptomatic population? How is the asymptomatic population possibly different from the symptomatic population? So when it comes to COVID-19, asymptomatic positive patients have been found to be a significant portion of our positive patients. The table you see here summarizes very briefly 5 journal articles that attempted to quantify the proportion of asymptomatic positive individuals. Without going into great detail, the results of these 5 studies indicated that anywhere from 40 to 80% of all positive cases came from asymptomatic individuals. To be clear, that does not mean that 40 to 80% of all positive, that does not mean that 40 to 80% of all people everywhere are asymptomatic positive COVID patients. It means that of the positive patients they looked at in these studies, 40 to 80% of those positives were asymptomatic. Many of these studies are unique because the patients themselves had a specific reason to be tested other than symptoms or even suspicion. In a way, it was kind of suspicion. So 2 of these are cruise ships, where every single person was tested on the cruise ship, staff and passengers. One is a long-term care facility, where there was an outbreak and every single person was tested. In the long-term care facility example, they were actually tested serially. They were tested over and over a period of time. The last one listed is actually a prevalence study where random population screening in Indiana was performed to determine prevalence. Several of these studies were outbreaks or hotspots. It is these asymptomatic cases that we are looking to find when we are looking to do asymptomatic testing. Often we're mostly going to be talking about screening, so workplace screening and school screening, situations in which we're looking to find and isolate outbreaks as early as possible in order to reduce transmission. These studies reveal that whatever the estimated disease prevalence is in an area could almost always be doubled if we were to include asymptomatic positives. The problem is that most people that are asymptomatic have no reason to go and seek out a test, nor should we be testing large portions of the population routinely. Instead, targeted screening of asymptomatic, especially high-risk groups, would have the most value. So now that we know who we are targeting and what portion of the population they could make up, what could be different about this specific population from a testing standpoint? So when we're looking at symptomatic versus asymptomatic populations, there are some potential differences in disease prevalence between the asymptomatic and the symptomatic. The asymptomatic population could have a disease prevalence that varies widely during an outbreak. It may match that of a symptomatic patient population if you're in a hotspot or there's an active outbreak that's really exploding. But without an active outbreak or a hotspot, it could be dramatically lower. We just don't quite know. And you did see from the previous studies that there's significant variation among all of those as far as the actual asymptomatic numbers. Because asymptomatic studies are so difficult to come by and quite frankly difficult to execute, we don't know all of these details surrounding disease prevalence yet. We also have the same or similar issues with distribution of viral loads as one of the big questions. So what I mean by distribution of viral loads is if the COVID-19 virus is in your body, where exactly is it? How is that virus distributed throughout your body? Your nose, your nasopharyngeal, your lungs. Where is the virus when you're asymptomatic? And is it in different places versus a symptomatic person? This affects our understanding of what the best sample type may be. We believe at this point in time that the best sample type is generally a nasopharyngeal swab. But there is data out there that argues for nasal swabs. There's data that argues for saliva. There are case studies where the nasopharyngeal swab was negative, but a sputum sample was positive. The ideal sample type is something that in the asymptomatic population, we just don't have a good picture of. So it's possible that we don't know what sample type should be used for the asymptomatic population. It might be different than the symptomatic population. We simply don't have a clear idea scientifically of why some people are asymptomatic while others are symptomatic. And then those symptomatic patients can also vary widely between mild, moderate, and severe illness. So without that understanding, it's difficult to know things like prevalence in an asymptomatic population, as well as the appropriate sample type. We do have several studies that are showing us that there may be similar levels of viral genetic material. This is really important when it comes to the limit of detection of a test. So early on, there were concerns about whether the limit of detection may need to be lower for asymptomatic individuals. It was hypothesized that asymptomatic positives could have less virus, and that's why they're asymptomatic. But so far, that's not what we've seen. We're so far seeing that they may actually have similar levels of viral genetic material, not decreased. So that's good news when it comes to limit of detection and the ability to use one highly sensitive test method on both the symptomatic and the asymptomatic population. But that also goes back to the importance of validation. So you need to do that full test validation in order to know the answer to that question for your population. So let's look at some asymptomatic testing strategies. If your lab is considering asymptomatic testing, in addition to a full and appropriate validation, your testing strategy should also be considered. This is especially true if you're looking to partner with your local community to support school or employer-based screening. So first of all, you want to look at which test should you use. There are two main considerations for choosing a test: sensitivity and frequency. There are definitely other factors that contribute to these two characteristics. A test with a high sensitivity may be more expensive and have a longer turnaround time, whereas a test with lower sensitivity may be cheaper and have a faster turnaround time, allowing for it to be used more frequently. Interestingly, a YALE study from July looked at these trade-offs. They found in a theoretical student population of 5,000 that by using a cheaper test that was less sensitive, less frequently, cheaper test that was less sensitive more frequently, they actually did every other day, so every 2 days, they could screen more effectively and reduce the potential impact from any false positive results that could result from that lower sensitivity. This table here shows what that higher frequency actually does. And this is just increasing the frequency by 2. It's not every 2 days. But the idea is that 70% sensitive test, which is the theoretical sensitivity of the test that Yale used, and then you repeat that test 2 days later. Well, now with the combined answer from both of those tests, your sensitivity is 91%. The good news is none of our tests currently on the market, so I'm talking the waived antigen, slightly lower sensitivity test, cheap, fast, between 12 and 15 minutes, have a sensitivity as low as the Yale example. So these are the actual sensitivities of our current EUAs for antigen tests. We have the Quidel SOFIA at 80%. We have the BD Veritor at 84%. And we have the newest one, let me get this name right, Lumira DX at 97.6%. So you can see that by repeat testing on these cheaper methods, the combined sensitivity dramatically increases from 80% to 96%, from 84% to 97.4%, from 97% to 99.9%. So serial testing and repeated use of less sensitive tests can be a solid testing strategy for the asymptomatic population or for using tests in these screening situations. So with all of that in mind, let's get into the nitty-gritty. So we are still going to cover the validation requirements for an FDA EUA, simply because the EUAs are already out there. We do have several labs that have received these EUAs. There are other labs that are in the process of receiving an EUA, and as we mentioned before, we don't yet know the implication of the HHS announcement. So we want to make sure that everyone understands these EUAs. Additionally, even if your lab chooses not to pursue an EUA because you no longer have to and it's now optional, this is still a good option as far as creating a framework for what your validation could look like. So what are the validation requirements for obtaining an FDA EUA? The FDA has divided their recommendations into two parts. The one you're seeing here is for a previously unauthorized test. The other option within their molecular template is for a previously authorized test. So in other words, if your lab is using a test with a current EUA and simply modifying it for use in the asymptomatic population, then the validation requirements for a previously authorized test are the ones you would need to follow. If you're creating an entirely new test and you're developing your own and intending to use it for an asymptomatic population, your validation should look more like this. So what they're looking for is at least 20 positive samples collected across at least three geographically diverse sites. And then the total number of samples required could be different. Basically, you'll need as many samples as it takes to actually get to those 20 positives. So it could vary based on whether or not you're in a hotspot. It could vary based on prevalence. It could vary based on your geographically diverse sites. So you'll go out there and you'll need to collect samples until you make it to 20 positive samples. There is also the possibility to use archived samples rather than this more active, prospectively collected. Keep in mind, this is a validation, which means for every single patient, you will need to screen for symptoms. Because again, you need to define that this is an asymptomatic patient. You will need to get at least 2 samples. So you'll need the sample you're looking to validate as well as a nasopharyngeal swab, if possible. Nasal swabs are also acceptable sometimes. And then you'll need to do the test on the method you're looking to validate through the EUA, as well as a comparator method. And that comparator method will need to be on an EUA method with high sensitivity that's been proven with that particular swab in that particular population. So it gets very complicated very fast when you're making sure your comparator method is sufficient. With that said, it is important when you're pursuing an EUA to go ahead and contact the FDA effectively early and often. So they have a template email. It's cdrh-EUA-templates@fda.hhs.gov. That templates email is where you want to submit any questions and get in with a reviewer as soon as possible, because they can help you create the exact validation and the study that you need to really smooth out the rest of the process, so you're not having to go back and rework anything. As far as thresholds are concerned, the FDA is looking for a positive percent agreement of greater than or equal to 95% and a negative percent agreement of greater than or equal to 98%. So as a reminder, PPA is sensitivity, so sensitivity greater than or equal to 95%, and NPA is specificity, so specificity greater than or equal to 98%. Although they have stated that they are willing to consider other tests that do not meet those thresholds if they meet other market requirements. So generally this has meant if it has a faster turnaround time, if it is a cheaper test, if it does not require an instrument, so if it's cartridge-based, for example, then they will look at that within their priorities, so that if it meets a need in the market, then they may not necessarily stick to these thresholds. So again, getting into that open conversation with the FDA early is going to be really important. The FDA is still effectively backlogged. They have been doing a lot of work. They have well over 200 EUAs that have been issued, but there is still a significant number in the waiting list. So this could be a long process. With that said, the guidance out there is that once your validation is complete and you can go ahead and begin testing even before your EUA has been received. So going back real quick to that previously authorized test, again, this means you're taking an EUA that had already been given. Maybe it's a commercial EUA, maybe it's a laboratory developed test that you yourself have already received an EUA for, and you're modifying it for the asymptomatic population so that you can make those claims about asymptomatic testing. So in this case, there is a minimum number of negative specimens. That's going to be your biggest difference. On the other one, it's as many total specimens as it takes. In this case, it's at least 100. There are a few other minor differences, but that's the biggest one, is you're going to need at least 100 negative specimens, as well as that same 20 asymptomatic positives. Now, as far as CLIA is concerned, we are always under CLIA. So you always want to keep those CLIA requirements for validation in mind. CLIA rules do require that accuracy, precision, reportable range, and reference intervals are assessed. but the CLIA requirements do not set any minimum requirements for validation. That's up to your laboratory director. So it's the responsibility of the laboratory director to establish the individual laboratory criteria. So that includes the number of samples that you would need and includes threshold criteria. Maybe that looks like the FDA validation. Maybe it looks a little different based on what your laboratory director wants. I'm also going to put a quick plug in here for CLSI, the Clinical Laboratory Standards Institute. They have a plethora of documents. Right now, they're offering several of their documents for free. What CLSI does is it looks at the CLIA requirements. It brings together industry experts to write in an easy-to-read format CLSI recommendations. And so you can use these to go through things like creating a quality plan, managing your document control, in this case, method validation. So the CLSI documents that would be applicable here, there's a lot of them, but just a few. MM03, Molecular Diagnostic Methods for Infectious Diseases. MM14, the Design of Molecular Proficiency Testing, External Quality Assessment. MM22, a microarray for diagnosis and monitoring of infectious diseases, and then several documents for method evaluation in general underneath the EP series. So if you have a CLSI subscription, I would definitely reference those documents in order to have a robust method validation under the CLIA regulations and the CLIA rules. So with all of that said, I do want to speak just a little bit about Acumen before we get into some of our Q&As and look at some of these poll results. Acumen is comprised of roughly 250 employees. We have diverse backgrounds that include operations, supply chain, finance, even clinical nurses and doctors. To date, we've partnered with over 500 hospitals to find opportunities for improvement and sustainable growth across the multiple layers of the clinical laboratory, as well as the imaging department. So today, we are profoundly impacting healthcare through these partnerships and through multiple COVID resources that can be found at acumen.com. I referenced a lot of studies here, so I've included those links. This PDF is available on the Big Marker website. So if you have a need or a want to look deeper into any of the studies I referenced, I have included them all here.
Thank you,
John, I will go
Kimberly.
ahead and hand it back to you.
Yeah, I appreciate it. Thank you so much for sharing that knowledge, doing all that research. I know there was a lot of reading and a lot of it coming in real time as we try to digest some of these changes. One other thing about Acumen, if you guys don't know, is their model is really interesting, where you can engage with them to just look at your spend on consumables, your equipment leases, all of those things. And if they're not able to save you money, you don't pay anything. They just take a portion of the savings. I know they've helped save hospitals and customers that I'm aware of a lot. And so they're really good at knowing what best pricing is on instruments, consumables, all of that. And so I would encourage you to reach out to them if you want to have an audit done or a review done of some of your spend there. And I know every lab is looking to find a little bit more funds so we can go a little farther. So it's good to have a partner like Acumen out there. I'll put in a short plug for Lighthouse and how we might be able to help as well. I saw in the poll that a lot of the groups are doing asymptomatic testing but are not validated for that. As has been demonstrated here, it might be good to validate for asymptomatic testing. If you wanted to go to the length of meeting the FDA's requirements for EUA, we can help with that. We have positive and negative samples that we can supply in order to do that validation, including positives from asymptomatic patients. Those are difficult to find right now. We're collecting positives, both nasal swab as well as saliva or oral fluid swabs. And so we're biobanking all of that just to help other labs to get set up to be able to offer this testing. And so if we can help, we do charge $24,500, and some of the services we provide along with that are laid out here, but we'll include the validation plan. We do all the analysis and can prepare and file that FDA EUA paperwork for you to get that accomplished, if that's something you want to do to kind of stand out from the crowd, or at least your customers know that you guys are doing things right. With that said, I would love to jump into a little bit of Q&A, and so I'm just going to give people an opportunity if they wanted any more information on kind of what we do to have that, but we'll jump into question and answer. I do see that several questions came in, Kimberly, through the chat function, and so one of those was regarding comments on the recent CDC change for asymptomatics. Based on what I'm seeing, I think that there's a little bit of a misinterpretation, but I want to hear from you as well, Kimberly, that I don't think the CDC was saying do not test asymptomatic patients as much as saying you do not have to test all asymptomatic patients, right? Just trying to distinguish between necessity there, and was trying to say that there's not a necessity for all asymptomatic patients to be tested. That doesn't mean that there are times where people are traveling or whatever it is, or maybe just want to be tested, that might be appropriate. But I would love to hear from you as well, Kimberly, on your thoughts on the CDC's announcement on asymptomatics.
Yeah, absolutely. So the CDC, the change that they actually made is to 2 questions specifically related to patients. If you have been in close contact within 6 feet of a person with a COVID-19 infection for at least 15 minutes but do not have symptoms, then it says you do not necessarily need a test unless you are a vulnerable individual or your healthcare provider or state or local public health officials recommend you take one. So that is more on the patient. It's definitely not anything saying labs don't perform the asymptomatic testing, nor does it say doctors don't order the asymptomatic testing. It's simply saying it's not required. You don't really need a test if you've been in close contact. The next one is if you do not have COVID-19 symptoms and have not been in close contact with someone known to have a COVID-19 infection, they're saying you do not need a test. Now, their next bullet says if you, and they put you in bold, decide to be tested, you should self-isolate at home until your test results are known. So again, patients, individuals could still make the decision to get tested, even though the CDC is saying it's really not necessary. Now, interestingly, there's a little bit of conflict between their different pages. So they have testing guidance kind of scattered all over their website. And so if you look into the testing for non-healthcare workplaces, they are still recommending viral testing of workers without symptoms as being useful to detect COVID-19 early and stop transmission quickly. They're also recommending testing for all close contacts of a person with COVID-19, regardless of symptoms. So their screening recommendations when it comes to workplaces haven't changed. So they're still recommending asymptomatic testing when it comes to workplace screening in order to reduce transmission. So they have a little bit of conflict going on right now. I'm definitely, with everybody else, looking forward to next week's CDC call. So that'll be Monday at 3 p.m. Eastern for those of you who want to jump on that to hear more directly from the CDC. But as of right now, as far as labs are concerned, I don't see the CDC guidance directly affecting our ability to test asymptomatic patients beyond the potential of reducing some of the demand from patients who would check the CDC guidance and maybe choose not to go get tested, whereas maybe last week they would have.
It sounds like most of the people in the audience here today are with you. 77% are saying they feel like asymptomatic testing is part of the solution, and 9% saying it is not, but maybe with the caveat that we just need to focus that testing on people with symptoms if you're in short supply of consumables or equipment capacity, and that makes sense as well. I think those points both have merit. We did have a question come in from Zach that wanted to know, are all asymptomatic patients that test positive actually infected with the virus? And so I'll let you respond, Kimberly. I think maybe he's saying even beyond false negatives, false positives, but what are your thoughts in response to Zach's question?
Well, there's definitely false positives. We've seen several examples of that in the news recently. We saw a significant number of NFL players come up positive over the weekend and then immediately test negative. We've seen a significant, I know I live in Ohio, so Governor DeWine recently had a false positive. So there's always that false positive risk, and the best thing that you can do for that, I'm seeing another question about false negatives that could be due to a viral load below the limit of detection of the assay. So I'll go ahead and grab both of those at the same time because they're related. So
Great.
If you have what could be a false positive, it absolutely could be because the viral load is below the limit of detection of the assay. So Ryan is absolutely correct. If it's below the limit of detection of the assay, repeat testing on that same method is not going to help you. However, repeat testing on a different method, for example, a more highly specific molecular method, will. So what you can do is you can create a testing strategy where you have reflex testing on your positives with a molecular test. So then you can have a more highly sensitive test that has that lower limit of detection, and then you're repeating a positive, so you shouldn't have hopefully very many positives. So you'd repeat those, you do some reflex testing to verify that they are not false positives. Now, maybe that testing takes a few days to come back and that person ends up in quarantine for a few days where they maybe didn't have to be. But in this case, that's what you want. You'd rather quarantine somebody for a few days that didn't need to be and then release them early than have a false negative where you have somebody with the virus running around thinking, okay, they're free and clear because we're negative. So that would be the strategy that I would consider, is that reflex testing. In fact, those antigen tests, they're all, there is some capacity for reflex testing those with a molecular test, depending on your medical director, your population, and what's going on in your lab.
Yes, great points. We have also a question about kind of what is the gold standard. I'll give my thoughts and you can add in any that you have as well, Kimberly. Is there a gold standard type of test or technology methodology that's generally accepted across the industry? It's a really good question. I know I have my thoughts are that the PCR test is the best for detecting actively contagious people or infected individuals. We do get a lot of calls that come in from groups that want to use the antibody test. And I feel like, what we do for a living is start and build labs, and I'm having to convince a lot of groups, especially outsiders from the industry, that they should not build the lab that they want to build because they want to build antibody testing labs, but they want to use that for screening purposes, right? And so they want to use those tests in order to clear people to come back to work, for people working at nursing homes, et cetera. And the appeal of it is obvious of why, because it's a cheap test, it's a point of care test, it's easy to deploy, the results are quick, inexpensive to operate. So all those things drive people towards wanting to use that solution. However, it's not a great tool for that, or even for individual diagnostics, in my opinion, more for contact tracing, population health type reasons maybe, or tracking infection. But you're not going to show up positive on an antibody test when you're actively infectious. And so, or there's a good chance you won't. And so I think the PCR test is the gold standard if that's what you're trying to accomplish. Kimberly, do you have any thoughts on what the gold standard is?
I think the PCR test is the closest we have right now. Because it's so new, we don't have a true gold standard. It's simply because it's been around nearly a year now, I'll give it that, but in scientific terms, that's not very long. And so there's still a lot of unanswered questions, things that we're just not 100% on. The sample types I was discussing is one example of that. So for now, PCR is the best we have. Normally, something like a viral culture would be even more of a gold standard, but that's not something we've been using for COVID. It's not something that we have an emergency use authorization for, and it would require a BSL-3 lab, so it's not a good option. So I would say the closest we have to a gold standard is reverse transcriptase PCR, but unfortunately, it's one of those things, it's the best we have available to us. I wouldn't go as far as calling it a gold standard.
Thank you. We have a comment from Ryan is saying, thank you for addressing this. The YALE calculation is a bit dubious frequent testing, low sensitivity tests will not likely be as robust a strategy as they suggest. I think one of the other concerns we had when looking at that YALE test, and there are some really neat possible tests that are coming out soon, and so we're investigating all of those, including extractionless systems, using saliva. Seems to be maybe the next wave of testing technology that's going to come out, and that would make it much easier to collect a sample, and if we can lower the cost enough that regular testing of people is a little bit more affordable and doable, I think that's a huge win. The YALE one, I believe they're looking at only one, I guess this is DNA marker versus the 3 that is kind of more typical, and that there's some concerns about that. I don't know if, Kimberly, you have any thoughts or comments to add based on Ryan's comment.
Sure. So Ryan's comment is about the YALE calculation, the study itself surrounding the theoretical student population. So the YALE test is a slightly different issue. I do agree the YALE test is only looking at one probe or primer, whereas most of them are looking for at least 2, one conserved region and one region that shifts a little bit more. So only looking at one region is a little bit risky as far as the test is concerned. I definitely agree there. For the calculation itself, more frequent testing with a cheap test is definitely a solid strategy. However, to like Dr. Radilovich's point, false positives can absolutely be problematic for the physicians that are receiving them. It can make it difficult to interpret. I know there's, let's see, it looks like Fred also threw in how do you know what are the possible false negatives to be retested for? So there's a lot of issues with effectively any result that's not correct. So that more frequent testing on a less sensitive test does give way to a lot of those issues. So I would only personally introduce that strategy if I had full control, meaning that the physician that ordered the test, anybody who would be giving advice to the patient, however you're managing your screening, that you have full control over that, so that you can work directly with those physicians on when to act, what your testing strategy is going to be, when to do a reflex test versus when to repeat. If you're having multiple physicians that are ordering these and you're getting patients all over the place and you're sending results back out all over the place, any kind of frequent, less sensitive strategy could result in a lot of problems in an outpatient setting. For identifying when you should repeat a result you believe to be a false negative, that's going to be down to now we're going to get away from asymptomatics here, but if there is, say you have somebody who's a caregiver for somebody that's in their 80s and they were around a COVID-19 contact, so somebody who later tested positive, and they were literally had that person cough in their face and they remember this action, no masks, no social distancing. If you have a reason to have a high suspicion that they definitely got something and you got a negative result, that may be something you consider retesting. Additionally, if you look at the CDC's website, they go through false negatives by day. So it's possible that this would be an option. On the slide said serial testing. So this may be a situation in which you may want to do serial testing. So maybe every few days you do another test, because the rate of false negatives goes down for a period of time and then it's going to go back up. So you're going to have a window in there where you're likely to get fewer false negatives. So if you have a reason to suspect that this particular person, that negative may not really be negative, maybe they're now developing symptoms, whatever the case may be, that would be the time for some repeat testing. That would also be the time for some serial testing. Do some testing at very specific time intervals so that you can catch a false negative, essentially.
Thank you very much. Yeah, that's great. And I think you also tackled the serial testing piece, which Tommy had a question about. David states that real world false positive rates is 0.8% And so that's good information. I'm not sure the source on that, so I'm sure that David did his research, but it is it's a fairly small number, but significant to those people that fall into that category. There are some other questions about, I don't know, the LOD for asymptomatics. I don't know if Kimberly, if you feel comfortable tackling that. I think we also have Dr. Magnani maybe on the line that could tackle that, or we might have to take it offline, but they wanted to know, Christina wanted to know if the asymptomatic LOD is different from the testing for symptomatic patients.
For me, from my perspective, there is nothing that I found within the FDA's requirements or within the studies I was doing that indicated a different limit of detection. Like I said, it appears that the viral loads are similar between the two populations, but I am not in a position to be able to go beyond those statements. So if Lighthouse has more to add, or if we need to circle back, do a little bit more research and come back, we can do that.
Yeah, I agree. That starts to get into the weeds a little bit. We'd be happy to have one of our molecular PhDs go into that if somebody wanted to really get into the science and engage us to look at a validation study. EJ also had a question about kind of how our validations work. We might need to take it offline, EJ, but we do provide the validation plan. We do provide the samples, the positives as well as the negatives, and those are asymptomatic positives that are included in that package. And then we do the analysis and preparation of that validation report to FDA EUA standards as part of our package. And so if you're interested, happy to talk to you more offline on that. We'll take one last question if that's all right, and it's about the upcoming flu season, concerns for asymptomatic individuals due to low viral load. Will COVID-19 antigen on the SOFIA 2 for point of care be the way to go? I don't know if you have any thoughts on that, Kimberly. I don't know if I'm going to be able to really speak to the SOFIA 2. I think I did hear that good old SOFIA originally was prone to some false negatives, I believe, where they were missing maybe 1 in 5 of the positives. And so still a maybe valuable test at 80% if you were to do some regular testing, but definitely some concerns with that as well. I don't know if SOFIA 2 fixed that problem or not. Do you have any comments on that, Kimberly?
The one comment I do have on that is the SOFIA is actually one of the lower performing antigen tests. So it has an 80% to an 87.5% sensitivity. It does have a 100% specificity. So when you're looking at the positive predictive value, so someone mentioned that earlier, as opposed to positive percent agreement, the positive predictive value takes into account the prevalence, the disease prevalence within the population. So positive predictive value is highly linked to specificity, and the SOFIA does have a 100% specificity. Once you have that 100% specificity, your positive predictive value goes way up and stays there regardless of your prevalence, your disease prevalence within the population. So I do think that there is a place for the SOFIA 2 with the upcoming flu season. I would absolutely work very closely with your lab through the validation to determine if any kind of reflex testing may be needed or even wanted. So as you go through and do your own validation, you're going to come up with a sensitivity and specificity that's a bit different than what the manufacturer has in its instructions for use. And it's going to be based on your patient population, your testing personnel, your exact reagents, and your exact instrument. And it has to be done for everyone at every testing location. So when you do all of that, I would take a look at your sensitivity and your specificity and make some determinations on, is there some reflex testing needed? And really communicate how reliable those results are, especially to your outpatient providers. So you want to make sure there's a good understanding. The last thing you want is an outpatient provider that believes that every result that comes across their desk is wrong. You also don't want a test that's going to do that to them, right? So you want to make sure that you are communicating and that the lab and your providers are in sync so that the providers know when to trust a result and when they might need to do a repeat test or question of results. So all of that is going to be really important. But like I said, it is 100% specificity, so that's going to be really important moving into the flu season.
Yeah, that's great advice and a great point. Thank you very much for your time, Kimberly, and for sharing that knowledge with everybody. Hopefully everyone leaves here today a little bit smarter. I know I do, and so I appreciate all the research you put into this. Did want to call attention to everyone about part 2 of this, where we're going to get into that FDA change. We'll digest that a little bit more, hopefully have some more responses from some of those government agencies to go over, and we're in the process of getting some higher level people from CDC and HHS to be on that call and to give their insight and opinion on what this means. And so join us. That'll be a week from today on the 2nd of September at this same time, the 1 to 2 o'clock hour, so you can have your lunch while you digest some knowledge as well. And you can register via that pop-up that I just put onto the screen to join us for that next webinar then. So thank you guys all for your time. I really appreciate it. Thank you, Kimberly.
Thank you.